Thank you. Let me see if I can do it this way. Is this better? Oh, much better. Okay, you can tell this is our first run, can't you? I forgot to use the microphone. Okay, so once again, my name is Dr. Carrie Schroyer, and I'm the Dean of STEM. And I'm so glad you're here to kick off our Community Connections event launch with us. So when we look at this, what is the goal of the Community Connections program? And I wrote it right up here for you, so we're going to go to that slide right now. Next slide, please. One more. One more. We're so close. One more. Oh, thank you. So why did we create this program? Because we want to enhance, oh, can't do it. Okay, I'll go back here. I'll look at it right here. To enhance technology awareness and AI and data science literacy by curating engaging speakers and free events to bridge knowledge gaps, inspire lifelong learning, and foster a more informed and connected community. And that's really important. We want everyone to have access to technology and AI and data science and see how it's affecting them. And this is one of the ways that Edmonds College is doing that. And we're so glad you're all here. So I'm very, very excited to announce our first speaker, Dr. Semih Tareen. Come on up, Semih. Okay. He has quite a background. He earned a bachelor's and a PhD from the University of Washington. And Semih, remind me again, it was molecular and cellular biology, right?
Yep.
Okay. He was a former executive director of viral, I think I might say it wrong, of viral vector research at Sana Biotechnology. And he has lots of experience previously before that. So he's also really committed to increasing science education and access. And he has done a lot of videos on YouTube, particularly during COVID. He was just showing me one of the videos that he did for kids. So if you have kids that are interested in COVID, I would really recommend you do a YouTube search for Dr. Semih Tareen. It's called Hi Ho, and it was so adorable, the way the kids were actually asking him about COVID. And he was talking to them in a way that they understood. So thank you, and welcome.
Thank you. Thank you. Thank you so much, Dr. Schroyer. Appreciate the invitation. It's an honor to be here with all of you. As soon as the slides are up, I'll start my presentation. Yeah, no problem. I know I'm all by heart anyway, so, but I'd feel bad for our audience to not see the visuals. As the slides are coming up, let me just tell you a little bit about who I am and why I do what I do. So I'm a virologist by training. For my PhD, I studied virus and host interactions. And when you think of viruses, because of COVID-19 recently, we often think of things that make us sick. But most of the viruses don't make us sick. In fact, most of the viruses on the planet don't even infect us. They survive with their host, and they propagate, and they do everything that every living organism on this planet, including us, are trying to do, which is propagate. We have to find a mate to propagate. They have to infect the host cell to propagate. We'll probably have maybe a few kids, and so every 20 years or so we'll be able to have a generation. Viruses will be able to have generations in the millions within hours. And so for that reason, technically, they are probably the more successful species. They have been around for about 1.5 billion years based on estimates. And they probably will be around after the human race is gone as well. And so I use that knowledge of how viruses are doing this for the purposes of curing illnesses like cancer, like diabetes, like lupus. And we do this with a type of therapy called gene therapy or cell therapy. Whether it's gene therapy or cell therapy, and I'll elaborate on what exactly that is, we need genetic information to be transferred, and viruses evolved to do that. So that's why we choose to use viruses, because it's already a tool from nature that works really, really well. And what you need is virologists like myself and my team who are able to understand the genetic sequences of viruses, chop away the dangerous bits. So for example, we use HIV, the virus that causes AIDS, for cancer cures. And all the dangerous bits have been chopped away, so it doesn't cause disease, but it does exactly what we want it to do, which is genetically transfer the information. The information is actually a very short genetic sequence. It's about 1,500 base pairs, and that's not very large in the grand scheme of molecular biology. And you literally just chop up those pieces and put them together and stick it into the virus and then give it to the cells of interest. We do this with immune cells because we are trying to educate the immune cells to recognize cancer and then to destroy cancer. And in my talk today, I'm going to give a little bit of a background on this topic. And then I'm also going to tell you, because AI is a very valuable tool, especially for the STEM program here at Edmonds College as well, a little bit about how AI is incorporated. Now, just a disclaimer, I'm not a computational biologist, so I personally do not do any data mining. I personally do not do any machine learning of the sorts, but I work with colleagues who do this. And so I'm going to give you an introduction on how they think about this topic and how AI is used for these technologies that we're going to cover today. And we're still waiting, right? Yeah, okay, great. No worries at all. Yeah, no worries at all. Let me tell you a little bit about this vacation I took last week. So I went to Miami. I love music, and I wear a lot of band shirts because that's one way that bands get supported is if you actually buy their vinyl or t-shirts. So I was on a cruise, and I don't normally go on cruises. I'm not the cruise kind of person, but it was this heavy metal cruise called 70,000 Tons of Metal. And it was great. It left Miami, it went to the Dominican Republic, and then came back. And there were 60 bands on this boat. And the schedule, I kid you not, was from 10 a.m. to 5 a.m. the next morning on 4 stages. And it was a blast. It was really fun to be there with about 3,000 or so crazy metalheads like myself. I told that story a little bit, and I dress this way because this is how I go to work as well. In a lot of science communication, one thing we try to get across is you don't have to be an old white guy to be a scientist. You know, we all love Bill Nye the Science Guy, but the demographic of scientists are changing. And this is not the talk I'm giving today, but I do give talks around scientists. I talk about one female scientist who's actually a professional wrestler, and she does wrestling as her hobby. I talk about, there's this famous band from Finland called Insomnium that I like a lot, and they're a world famous touring band, and one of their guitarists, he teaches evolutionary biology at Cambridge University, etc. So I try to give all these examples of different scientists from different backgrounds, and it'll always get a kick out of that. Yeah, so the best metal band I saw on the cruise, I was really excited to see this band from Germany called Lord of the Lost. And one of my guilty pleasures is the Eurovision Song Contest. Have any of you heard of the Eurovision Song Contest? There's even a Will Ferrell movie. It was Will Ferrell, wasn't it? Yeah, and it's hilarious. So it's this European Song Contest, kind of like the World Cup, but mostly European countries. Australia's in there too somehow, probably because of the Commonwealth. And they all submit a song, and then in May, there's this final of all these songs, and all these songs are all over the top, and they're stage performances. So Will Ferrell made this hilarious movie called Fire Saga on Netflix about the Eurovision Song Contest. So anyway, so Lord of the Lost was Germany's submission last year for the Eurovision Song Contest. And they're a metal band, but once in a while, countries will submit a heavy metal band like this as well. And once in a while, they will win, too. They didn't win last year. I wanted them to. But it was great to see them live. I had never seen them live, and it was fantastic. Great stage show. Yeah. All right. Yeah. I'm going to get a sip of water real quick from here. Flicker. Say again? Oh, of course I am. Yeah. Yeah. Yeah.
How's that? I'm sorry, I'll move that too.
That's great. No worries.
Okay, thank you. Okay. Don't worry, he's coming back.
I saw these things, these mini projectors you can buy that connect to your phone, and then you can project it off your phone. I wonder when the day would come where they would be present in everyone's house, so you can just say, hey, here's my 7,000 tons of metal, heavy metal cruise that I just did photos and the presentation. Yeah. Yeah.
Is that okay?
That's okay.
Are you sure?
Yeah, absolutely.
All right. Let's just give a round to Dr. Semih for being such a good sport on this. Thank
You're very
you.
kind. Thank you. This
All
is this kind of stuff happens all the time. You know, it's I really appreciate all of you who have come together for today, and all the staff who's helping us, the technical staff especially, because these things happen all the time. I mean, you've probably been to shows where the band is awkwardly just waiting on stage, and there's probably a simple line somewhere that's not quite working, and that's the solution. And anyway, so and by the way, on that cruise, it was amazing to see. All of them had their technical staff with them, the sound people, the lighting people. So thanks to everyone, including the team above there, all this happened. So I really appreciate it. So one of the videos that the professor was mentioning, if you YouTube Kids Meet a Virologist, it's this channel called Hi Ho Kids that has about 5 million subscribers, and Kids Meet a Virologist has over 1 million views right now. They asked me to talk to children. It was during COVID, so it was over the internet, over Zoom. And it was adorable because these kids had so many amazing questions about what the virus is, what's going on in the world, and I was able to perhaps shed even just a little bit of light on them, and that was really, really fun. So you could check it out in your free time. I'm so sorry. Yes, so what kind of questions they asked were everything from, first of all, we started from, hey, so do you know what I do? And a lot of them thought I was a hairdresser because of my hair. And then I'm a virologist. I explained to them what that meant. And then from there, we got into talking about things like, what is COVID-19? And then why are we at home? Why aren't we at school? So explaining how the virus spreads and so on. It was adorable. A lot of them knew quite a lot, actually. There was this one girl who was just filling in all my words, and she was a step ahead of me all the time. It was phenomenal. What was your question? I'm sorry. Oh, yes. It was absolutely adorable, yeah. Great, okay, wonderful. Yeah, that's completely okay. You know, I wouldn't mind if I can have the laptop back because of the order of slides and what to cover. And if you're still working on it, yeah. I'll go ahead and start. Please. So, as I mentioned, I use viruses to cure cancer, diabetes, and lupus. And throughout my talk, I may say I or I may say we, but just so you know, it's never I. It's always a team of people, and it's always my colleagues as well. So my team alone can't get this done. And it is also decades and decades of research and iteration and errors and mistakes and iteration and so on that allows all this work to happen. So we had a drug that was FDA approved in 2021. The drug is called Breyanzi. That's the brand name. Every drug gets a fancy brand name so that you can have all those silly commercials on TV. And then every drug gets a generic name by the WHO. The generic name for this is lisocabtagene maraleucel. And the World Health Organization names drugs based on categories. So this was a gene therapy and a cell therapy, so the words gene and cell are in it. So lisocabtagene maraleucel, there's the cell at the end, and lisocabtagene, there's the gene in the middle. We just used to call it lysocell for short. This is a kind of cell therapy where the patient who has cancer, usually blood cancers, they come in, we apherese the patients, and we isolate very specific cells called T cells. And those T cells are a type of immune cell. And then in the lab, we send the patient back home, or if they're in the hospital, back to their room. In the lab, we take those T cells, and then we add the viruses that my team and I have been preparing. We call these viruses viral vectors because viruses imply something that might make you sick or something that spreads. These don't spread. They are a dead-end product. So you add them to the cells, and they deliver the genetic information. They enter the cell, travel through the cytoplasm, enter the nucleus. They integrate that genetic information into the nucleus, so they're part of that patient's genome in that one cell. And they do this across hopefully millions and millions of cells because we're adding this to a pool of cells in a bag. And then we grow these cells over a week or 2 or 4 weeks until they reach the ideal number. When they reach that number, we check these cells to, first of all, make sure that gene delivery event has worked, and to make sure that they express that very special protein. Thank you so much. I really appreciate it. Thank you. That they express that very special protein. Now, you may remember DNA makes RNA, and then RNA makes protein. So the gene delivery accomplishes a protein expression. That protein is a very special kind of protein. It's a synthetic protein. We took chunks from nature. We took, say, proteins that are on the outside of cells. We took proteins that are on the insides of cells, and we put them all together. And they're supposed to serve 2 purposes. Number 1, recognize the tumor. And number 2, to kill the tumor. In the intracellular part of these proteins, there are special structures that signal the cell to secrete these lysing enzymes. These lysing enzymes then will kill the tumor, and that's how they work. So we then call the patient back, and then we deliver these cells back into the patient. Now, these patients usually stay in the hospital because they could get really sick. Why? Because they could end up with what's called a cytokine storm. Cytokine storm is, so for example, I get the man flu a lot where I'm on the couch and I feel so bad, I can't even play Xbox when I'm sick. And that's because I have the so-called man flu. And what that is that's my body fighting against the infection by releasing cytokines. And that inflammation of cytokine release is exactly what my body evolved to do. And it does it well, but it makes me feel horrible. So that cancer therapy, imagine all of a sudden these patients get millions of cells that recognize tumor and kill them in the blood, just like that. And they release all these enzymes. And that creates that cytokine storm, which is why these patients can get really sick. They often end up in the intensive care unit after this therapy. Rarely, not always, but some of them end up with what's called neurotoxicity, where their brain starts getting affected. They become catatonic, they can't even talk. Now, this sounds horrible, right? Well, guess what? After all this, especially if you can manage all those symptoms in the ICU, thanks to the nursing staff and the doctors, all that goes away, and these patients all of a sudden start getting better. The very first poster child for this therapy is somebody called Emily Whitehead. You can Google her name. Emily Whitehead was, I believe, 6 years old at the time. She was diagnosed with a kind of lymphoma leukemia. And she was one of the first pediatric patients who received this experimental therapy. She got very, very sick in the ICU, just like I mentioned. And then her doctors thought of one thing. They thought, we should suppress that inflammation a little bit. And they gave her special drugs that were able to recognize that inflammatory response and suppress it. Guess what? Today, during COVID-19, one of the approved therapies, if you're very seriously sick in the hospital, is a similar kind of immune suppressant. It's called dexamethasone. And it's used in severe COVID-19 patients because that cytokine response, that's the response that damages your lungs. It's not the virus. The virus doesn't kill your lungs in COVID-19. It's your immunity kills your lungs. So in severe patients, you will give dexamethasone to suppress that immune response. Same in those cancer patients as well. So her doctors gave her that drug, and she survived, and she has been cancer-free since 6 years old. She's now 18, I believe, and she just started college this last fall at that same university where that trial was conducted at. So she became the poster child of this story. If you, in Science magazine, she was on the cover, in the New York Times, et cetera, et cetera. Now, it doesn't always work like this. The success rate is about 50%. So only in about half of the patients who get this therapy are they cancer-free for life, or for life meaning the duration of the study. So the reason it doesn't work, the remaining 50%, they often have to get a second therapy or they have to try something else. And we're still all trying to figure out why. So my team and I, we designed that virus that delivers the gene, and we also designed that gene. And as we do this, we rely on just years-long knowledge. We actually don't really use anything fancy. We rely on antibody sequences that have been developed to recognize tumors over the years. So we just take one of those and we engineer it into our virus. And then we take other chunks of information from all over the years, and then we just assemble this chimeric protein. And for that reason, it's called a chimeric antigen receptor, CAR, CAR for short. And these are T cells. So if you want to read more about this, this is called CAR T cell therapy. And there's a lot of great information on the web, and you can find out a lot of that when you take a look. So CAR T cell therapy, check it out. Now, what we are doing right now is the process I told you is very cumbersome. The patient comes in, we take their blood cells, we give them the virus in the lab, we grow those cells in the lab, and then 2 to 4 weeks later, they get the cells back. This 2 to 4 process has to happen in a very special GMP lab. GMP stands for Good Manufacturing Practice. We built one of those labs in Bothell, and that's where it's done, and under very strict requirements. Now, sometimes the patient's cells don't grow very well. Sometimes they get contaminated. And we have to give the patient a call and say, sorry, you're not getting your cells. And a lot of these patients, keep in mind, have tried everything else, and this is their last line of treatment. So this is horrible news for them and their families. What are we doing to get around that? Well, right now, we're trying to engineer viruses that we can give directly to the body. So instead of doing this in the lab, we would give, just like HIV, people catching HIV, and HIV infects T cells in the blood, we would give viruses derived from HIV, but they're safe, to the blood so that they would target the T cells in the blood and make this therapy, CAR T cell therapy, in the body. Not in the lab, but in the body. So what this means, we don't need a GMP facility, and we don't have to take 2 to 4 weeks, and everyone gets their therapy. We don't have to give anyone a call back saying, sorry, your cells didn't grow. So for that reason, to do this, we're working with AI and without AI. So the without AI part is just years of research. We are taking different antibody sequences that recognize T cells. We are adding them to our virus so that our viruses will infect T cells in the body like we want them to. And then we do all the experiments hoping this is specific. So we have to screen the body and make sure this virus doesn't go anywhere else. It doesn't go to the brain or lungs or liver or anywhere else where it would cause a problem. And most importantly, we have to make sure this virus doesn't go to the germ cells, so sperm cells or egg cells. And the reason for that is if this person is going to have children, and if that virus enters their germ cell, then what that means is their genetic information will forever have this viral sequence in it. And when they have kids, every single cell of that child will have that viral sequence. And when they have kids, every single cell in that child will have that virus. So we don't want this to happen. Now, this actually happens, believe it or not. All of us in this room, when the human genome got published in the 2000s, it turns out what we call genes are only about 1% to 2% of our genome. And the rest, people used to call it junk DNA. We didn't know what that meant about 20 years ago, but now we do, thanks to computational biology and more sampling and understanding of life around us. So it turns out half of our DNA, all of us in this room, is made up from ancient viral sequences where our ancestors, not our parents or grandparents, but I'm talking evolutionary ancestors over hundreds of thousands to millions of years have been infected by viruses in the germ cells, so in the sperm or egg cells, such that that virus sequence has become a permanent part of that DNA, and all of us are carrying it right now. So we don't want this to happen in a clinical setting. We don't want to cause this, and we control everything to prevent this from happening. But in an evolutionary sense, it has already happened. So all of us in this room are half virus for that reason, genetically speaking. And we actually see this in real life happening right now in Australia. Koalas are infected by a kind of virus. It's a kind of retrovirus. And depending on the coast of Australia, on the geographical map, you're seeing this happening in real time. So I talked about this happening to our ancestors hundreds of thousands of years ago. In the koalas, we're seeing it happen in real time, where in this part of Australia, their genomes do not quite yet contain this viral sequence. But in other parts of Australia, it seems like this has been happening on a few generations already. So that viral sequence has already become the part of their DNA. Imagine if all of us caught HIV, and if, well, actually not all of us, we don't need all of us to catch it, but if we caught HIV, and if HIV infected our germ cells and we had children, that HIV genetic sequence will be in every cell of our child's DNA, and then their child's DNA, and so on. So theoretically, one day HIV, if it ever did this, it doesn't, by the way, but if it ever did this, theoretically HIV could become a part of our genome one day. And that koala virus I mentioned, it's actually related to HIV. It's a kind of retrovirus, the same way that HIV infects cells. So I digressed, I know, but this fascinating evolutionary story is something we want evolution to take care of, but we don't want to recreate that in the lab. That's why we have to make sure these gene therapies are safe to the degree possible. These gene therapies are safe in a risk-benefit way. By the way, these viruses that infected us over the centuries, hundreds of thousands of years, they don't just sit there in our genome. They actually, some of them actually do something. And when our mothers were pregnant with us, there was a placenta that was formed, and that placenta had very important layers. One of those layers is called a syncytiotrophoblast. And syncytiotrophoblast is formed by several cells coming together to form this special layer, and those cells fuse and form that layer. And that layer is important for nutrition, for blood exchange, and for protection of the fetus. So it turns out a very special protein allows those cells to fuse with one another, and that special protein comes from a virus, but not from a virus in the environment. It comes from a virus in our genome. So it seems like, and not just humans, a lot of mammals do this, mammals, at some point in evolution, have co-opted these viral sequences, and they've evolved to use them in placenta formation and protection of the fetus. So much so that one of my favorite science writers, his name is Carl Zimmer, he has books, but he also writes for the New York Times. He said in 2012, if it weren't for a virus, none of us would ever be born. And at least in this biological context, that is true. So we want to make sure none of this stuff happens to shape the evolution of humans unwantingly, but we want to make sure that these CAR T-cell therapies are safe within the risk-benefit profile. I say risk-benefit profile because all these drugs get approved based on risk or benefit. We get vaccinated based on the risk-benefit ratio. You know, the benefits outweigh the risks. The risk of catching COVID is worse, et cetera, et cetera. So same here. These patients have cancer. They're going to probably die. So even though these therapies are risky, the benefit profile outweighs the risk. And of course, the MDs make these decisions. I'm not an MD. I can't make these decisions for them. All right. So AI comes into play when we want to target a specific cell. We're researching this the old way. We go to our lab, we read papers, we find these different sequences, we do these experiments in little tiny wells in the lab, and you can only do so much. You can do 12 wells, 24 wells, hundreds of wells, but now you're talking a lot of plastic stacked on top of each other and a lot of pipetting where you just do this all day. And There are machines as well that help, and they're expensive, so not all companies or labs buy those machines. So that's where you need people with good wrists to do that. And you can do it that way, and it will work, and it's just fine. So no AI is needed there. Of course, you have to experimentally validate it. But in AI, here's how companies and researchers use this. They take the knowns, they feed the known information into a model. So known meaning, we know that this antibody binds that cell. We also know this other antibody binds that cell. And let's say you look at 100 or so antibodies. You feed all that information structurally in a 3-dimension, or as a genetic sequence, or both, into the model. The model then learns from that algorithm. And there are several algorithms. Some work better than others. And then that model will give you completely different sequences to try. Sequences that may not exist in nature, sequences that maybe you thought of by chance, but the combinations, the possibility of you having thought that exact sequence is slim, because we're talking a combination of several sequences within hundreds to thousands of base pairs. And then, so that's what AI does. And then now you have to validate that. You validate that experimentally, but AI can also sometimes validate that in some kind of whatever information you feed it. So this works, and there's a company in Seattle called Shape Therapeutics, and Shape Therapeutics uses this. They use AI to change the outside of the virus, so modeling the 3-dimensional structure of their virus for better targeting, for specific targeting. They use AI to change the sequence of the genetic information inside the virus. So they use something called a guide RNA. A guide RNA does exactly that. It recognizes sequences. It guides things to other sequences in the cell. And you can guide things into a cell where there's a mutation, and you can fix that mutation. So they're trying to do this for the purposes of gene therapy for diseases that have been inherited where there are mutations. So they have to evolve these guide RNAs, and they use AI to do exactly that. They feed the knowns into a model. That model comes up with many iterations of what those guide RNAs can look like. And then they test these in a lab using robots and thousands of wells, so you don't have to pipette all of them. And based on that, they have successful hits. And if you have hits like that, that's a good thing, because first of all, as a company, you get to patent that sequence. And patents are important for companies because a lot of substantial seed investments are needed for companies to get started. And companies create value in many ways. One of them is through patents. And the second is they now potentially have something that they can try for the benefit of the patients that might help. So this is one example of how AI is used. Another example is in scientific imaging. So I have moles. I go to my dermatologist and I say, hey, this looks kind of bad. What should I do? Dermatologist kind of squints and says, oh, it's probably fine. Just see if it grows. So you can actually design software that is now used for medical imaging, whether it's identifying moles and determining whether it's precancerous or not, or looking at chest x-rays and CT scans, identifying if a haze on an x-ray is actually a cancer or not, or if it is COVID or not, and so on. There are examples of this, and they're actually approved softwares by the FDA for these to be used today. One of the images I can't show you right now, but it's an image of, they took radiologists, about 10 of them, and they had them diagnose an x-ray, and then they took the same AI software, had it diagnose an x-ray, and the conclusion was that the AI was pretty darn close to the humans. And it seems like it hasn't surpassed humans yet in terms of catching something that humans have missed, but that just highlights how amazing radiologists can be when they analyze these images. So quite fascinating. Another study that I've come across is for the entirety of COVID-19, so from diagnosis all the way to drug development, to prognosis, and then to the future of the epidemiology of a pandemic like COVID-19, you can use AI to model these and to get some kind of a predicted outcome. Keep in mind that in all these cases, it's a prediction, and keep in mind that it is only as good as what you've fed it, information you've fed it. So the information you fed it has to come from labor-intensive, hour-intensive studies that have been done by humans at some point. So without that information, and even today, I haven't yet used ChatGPT, but even today, ChatGPT is only as good as whatever information people have fed the internet that exists on the internet. So, this is not some type of a miraculous thing, but it is being used actively throughout research today. And there's one more topic that I want to cover real quick. In the cell therapy that I mentioned, the cell therapy cycle that comes in is the patient gets diagnosed, and the patient will have tried different medications before they come to us. And then we take their cells, as I mentioned, for the approved CAR T-cell therapies, and we deliver the viral vector to deliver the gene. We grow the cells in a lab, get the patient back, and give them the drug. And at each step along this way, it is from the patient's prognosis, it's from the quality of their cells, because right now what we do in the lab is we look under a microscope, we say, are they alive or dead? Okay, check. Do they express the protein we want? Yes or no? Check. We don't do any more complicated measures, but some of the measures that we want to incorporate are, if these cells end up back in the body, will they be able to kill? We don't really have a quick way of checking that. So AI software is being incorporated to look at markers on these cells to understand whether or not they'll be able to kill once they're inside. And then AI models are being developed for the dosing of the patient. As the cells are getting dripped in through an IV line, you can actually monitor the health of the cells and so on. And then ultimately, AI is being developed to see if the prognosis for these patients will be good, depending on the treatment outcome. The challenge here is, of course, not only that these models have to work, and research shows that so far they seem to be pretty good, but the challenge here is the regulatory authorities have to catch up. So in the United States, that's the FDA, the Food and Drug Administration. We work with them for all of our clinical trials to be approved. We work with them for our drugs to be approved. And for example, you will submit 1,000 or so pages of research and such just to be able to begin a clinical trial with 10 people and so on. But anytime AI is involved, you're now talking about a software that is going to make predictions, and based on those predictions, the medical staff is going to either change the course of treatment or assess treatment and so on. And then based on that, your insurance company is going to decide whether or not it will cover that treatment, because an AI said that you should take it versus, say, a medical. So all these things have to be figured out by the lawmakers, by the policymakers, and most importantly, by the regulatory agency. So there's this one paper that I came across where it seems that in Europe, the EMA, the European Medical Agency, and in the United States, the FDA, have gaps in being able to, today, being able to assess AI, because AI is such a fast-growing field, and biotechnology is such a fast-growing field, that by the time all these things are ready for the clinic, the government agencies, they've made progress, but not enough. So they've identified these gaps. And credit to the FDA. So I've been working with the FDA since 2010, where I have to be on the call with them to explain to them what these therapies are, to answer their questions on safety issues, have written commentaries back and forth. And my impression has always been they hired some of the best scientists on their staff to make these decisions. And for the sake of overcoming the paper burden and any kind of other burden, they're doing their best. In fact, recently, because cell and gene therapy is advancing so fast with so much potential, they have been able to address this fact by saying for cell and gene therapies, our approval process has to be fast and fast enough to make sure the patient is still safe, but to also make sure that patients are not held back from any potential therapies that might save their lives. So that's the end of my talk today. I hope I was able to give you a little bit of information, and I'll stick around for questions about what I do as a virologist. So I'm not so much about trying to cure infectious diseases. I'm not an infectious disease. In fact, I love viruses, you can tell probably. And nothing would make me happier than to see them thrive, and of course, without human disease. And we use those fascinating creatures for gene delivery. For example, last year, Duchenne's muscular dystrophy is a horrible disease that got its very first gene therapy approval. Beta thalassemia got its very first gene therapy approval. Sickle cell disease got its very first gene therapy. So, and this year is predicted to be the record in gene therapy approvals. And all this is happening thanks to viruses and or another technology called CRISPR. And you may have heard of CRISPR. It looks like these fancy scissors because they always draw it as scissors where they can cut pieces of DNA. But CRISPR, it actually comes from a viral defense mechanism. So bacteria also get infected by viruses. And bacteria have evolved defense mechanisms where they will recognize the incoming viral sequence and they will cut it with enzymes. So that's CRISPR. So we've taken that from nature and we now incorporate it in biotechnology as a way to specifically recognize something. In bacteria, it was the viral sequence. For our purposes, it's like a gene mutation, and cut it out. And this is going to be the fastest growing year yet for gene and cell therapy. I hope I was able to tell you a little bit about what cell therapy is. Patient cells in the body are changed genetically. These are mostly immune cells. I didn't talk about diabetes today, but we recently started a clinical trial at Sana Biotechnology where we take pancreas cells from cadavers. We deliver viruses to deliver genetic information to those pancreatic cells, and then those are given to diabetes type 1 patients with the goal of those cells secreting insulin and those cells not getting rejected, because just like organ transplants, cells from a different person will get rejected by your healthy immune system. So we have to figure out a way around that, and we deliver genetic information to do that. I didn't tell you about that today. I didn't tell you about our lupus clinical trial, and I didn't tell you about our Parkinson's clinical trials either. So that's for another time. But viruses do come to the rescue. And I told you a little bit about how we do this the old school way, just based on reading papers and a little bit of guessing, a little bit of trial and error, and it works. It's good enough, and good enough sometimes saves the day. But AI comes to the rescue where you have a limited set of libraries, and you can test them all, but that's it. AI can now help you predict confirmations that you didn't think of. You just have to make sure you feed it the right information, and that you use not just one, but several models, because some models are better than others, and then you have to validate them experimentally or still using AI, but you still have to validate them. And then finally, regulatory agencies have to catch up to all this, and I believe they will, because so far they've convinced me that interacting with them, they know what they're doing, and whether it's the agency or whether it's scientists like us, we have nothing but the patients and their families' best interests at heart. So thank you so much for your time. I really appreciate your attention today. Thank you.
So I just want to thank Dr. Tareen for being such a great sport. Thank you so much.
My pleasure.
It was awesome. Thank you. And I want to invite the audience to ask questions. Dr. Tareen graciously agreed to stick around for a little bit. And I'll start with one question. Did you always know you wanted to be a virologist?
Yes. I love that question because I get to talk about horror movies, and here's why. So I was born in '76, so I grew up in the '80s. I was an '80s kid. And there was this film called Cujo, based on a Stephen King story of a rabid dog, right? So I love that film, and I watched growing a lot of horror movies. My parents wouldn't allow me, but I had an awesome granddad who would sneak me in and let me watch him. And then, I didn't tell you this, but I had a slide about a little bit about my background, but I'm from Turkey originally. I was born and raised in Turkey. Turkey wants us to call it Turkiye today, so whatever. But I'm from the Aegean coast of Turkey, and over there, when I was a child, we had a rabies hospital. This sounds like it's a horrible place to be. It wasn't, but I think they still had enough rabies cases in a big city. It wasn't Istanbul, it's Izmir. It's a still relatively big city. And I had to go to that hospital, and let me tell you why. So Turkey is famous for its cats. If you ever go to Istanbul, there's cats everywhere, and they're adorable. They're all vaccinated, they're all taken care of, but they're feral cats, and everyone loves them. In fact, there's an awesome documentary coming to SIF Cinema soon. It's an older documentary, but they're re-showing it called Kedi, K-E-D-I, which means cat in Turkish. And it's about cats in Istanbul. It's an adorable documentary if you love cats. There was this cat. We played with it as a kid in the street. We loved it. It was taken care of by one of the florists in the neighborhood. The cat died. The florists had the cat checked. Turned out it died of rabies. None of us recall rabid behavior, but still, so the entire street of us kids, this is in the day when kids still played on the street, right? We had to now go get a rabies shot just to be sure, even though we weren't scratched or bit, etc. So all our moms and dads took us to this rabies hospital. And over there in a waiting room, there were about 20 or so people all there for a rabies shot. And there were stories of people getting bit by dogs, or people waking up and their baby's face was gnawed by a rat, and all sorts of. So I was a kid, and the love of horror really made my imagination just crawl in a way where I really freaked the hell out. And so Cujo, that experience, and then the last thing is this film from Turkey, actually. It was about a village in rural Turkey. There's a rabid dog. By the way, if you've ever seen the movie To Kill a Mockingbird, they have a rabid dog there too, right? It's kind of a cool part of the story too. So rabid dog, and the kids get bitten, and they all have to go to this hospital. But there's not a hospital in the village, so it's a bit of the film is a commentary about life in rural Turkey. So they have to trek through rivers and mountains to the nearest hospital where they can all get a rabies shot. And one of the kids decides not to go, and they had this really violent scene of this kid just suffering through rabies and dying. And it's a really horrible way to die. Rabies is the deadliest virus to humans today, still, if it's not treated. It's none of the others. It's pretty much 100% death guaranteed if you're not treated. There are very, very few cases where people weren't treated with a vaccine or antibodies, but they were put in an induced coma in an ICU, and they made it out. One of these is a very young, brave young lady from Wisconsin, Milwaukee, and she actually has her own YouTube channel about this whole experience. I forgot her name, but you can look it up. And for that reason, the treatment she was given was called the Milwaukee Protocol. And they've tried to repeat this in other rabies cases, and it doesn't work all the time. So it's clearly not a cure. So anyway, so rabies, so I digress. But that's why I knew I wanted to be a virologist. It was also the same time when the AIDS epidemic had started in the 80s, and all the flashy news stories, et cetera, at the time. I was just absolutely fascinated. And I didn't really want to cure it. I just wanted to understand how all this worked. And you can tell today I don't cure infectious diseases. That's not my specialty. There are amazing virologists who do that. Thank you.
Now, who else wants to be a virologist? It's not too late. Who else has a question? Okay.
Please.
Wayne.
This works too, so I'll just use this as well.
Thank you very much. I'm going to ask a question more on the human resource side with your particular specialty, being a virologist, but also in the medical profession research. As AI becomes more prevalent, more refined, will there be, at some point, I guess, I don't want to call it an opportunity, but do you see your position or others in the profession of research now being excluded from jobs because AI now is doing the modeling, or the modeling is still happening, but it's being done by AI as opposed to running through it experiment, multiple trials. So what's your opinion on that for the human resource side of your profession?
Thank you. It's a great question. I'll just stand here since this mic also works as well. Thank you. I'm not worried about it. However, there is the following fact, which is even in a biotechnology setting, you need liquid handlers. You need people who are pipetting and moving liquids around for experimental studies to be validated. The same concern that those people, and often these are people who are, I did this as well right after my bachelor's degree. These are often the first jobs you get after college or during college. And it's very labor intensive, and it's kind of like, we joke about it about, hey, we're just the monkeys doing the pipetting kind of thing. But in some parts it's true, and you need validation of those studies with some type of a liquid handling setup. So it's possible. We were worried that those jobs would go away when the robots came in, but it seems not everyone is able to afford the robots. So for that reason, those jobs still exist out there today, and some of you are probably even working those jobs, and or as undergraduates, you're probably thinking about them as well. And the part where some jobs could be taken over is AI is not costly. It's just some cheap software probably and some cheap hardware, because today computers are cheap. It's possible that a lot of the liquid validation would not be necessary someday, where these models become good enough, where we are all comfortable that the experimental validation is not needed. So now those liquid handlers, are they out of jobs? I don't know. It's also possible that, say, a startup biotech that relies on these AI methods may have a staff of 20 people instead of 50 people, simply because of your question being accurate, where there probably are some jobs that were unnecessary because AI with the humans is doing good enough and solving just the right amount of problems. So the answer, I think, therefore becomes yes for the liquid handling, and yes for even outside of the liquid handling and just thinking and so on. The part that would be really fascinating to me right now is, so I'm not in the lab anymore. I haven't been in the lab for about 10 years, but I run teams of people who are in the lab. But I work more on strategy and leadership for the company. So we try to establish a culture for the company, on diversity, inclusion, equality, as well as our values, as well as leadership and strategy. What I'm most fascinated by is there might come a day where AI could help us make decisions, because we have to make decisions based on very little data, very little scientific information. Why? Because if we wait to get all the information, it'll be 5 more years, and a lot of patients will die in those 5 years who could have gotten access to therapy. So we just have to find the right balance of safety information and scientific data to know that it works. And I think AI could help us there, because humans are emotional, and you have a spectrum of decision makers. I'm a very harsh decision maker. I love just making decisions and move on, and I'm wrong so many times for that reason. And then I rely on my colleagues who balance that with me, who are very calculated and thoughtful, and I would love to have software to help me out personally. And it could make me obsolete, and that's okay. I'll just be a musician or something if I had to. Thank you, great question.
Semih, çok teşekkür ederiz seminar için.
Thank you. That means thank you in Turkish.
Thanks for the seminar.
Thank you.
So I teach here in the biology department, and with some of our students I've seen, some of our students that I've had in my cell biology classes over here in the audience, and thinking about their future and how we can serve them, especially in introductory courses in biology. What are some of the skill sets and some of the just knowledge base that they're going to need in the future coming out of a program like this, or that in a way that we can serve them? What skill sets are they going to need, do you think, to be competitive in a future that's...
Thank you. Very important question, because we often aren't taught how to get a job. Maybe some colleges have those programs, we often aren't taught how to interview, and we often aren't taught how to survive in say, a biotech setup. There are some places that try to bring speakers like myself and others to talk to students about these things, but we aren't taught a lot of those things. So I do think complementing a strong science education with real-life skill education that is relevant to biotechnology would really go a long way. So I'll give you one example. When we interview, we, of course, look for scientific skills and helping students understand the best way to present their thinking, the best way to present what they know, and to think on their feet is a great, so that's one, what I would recommend. And for them to do that is to practice giving talks of either research projects they've done or of a scientific topic, and then to have Q&As with them so that they get used to this. So that's number one. And the second thing they have to do is what's called behavioral interviews. This is common in a lot of HR settings or in a lot of job searches, but in biotech, it didn't used to be common. And now it is, because biotech is catching up from other tech fields where they realize behavioral interviews really tell you who this person is. Questions such as, give me an example of a time when you made a mistake. What did you do? How did you handle it? Give me an example of a time when you disagreed with your supervisor. How, why? And based on answers, a lot of insight comes out. And I find that a lot of recent graduates have not been familiar with this type of a setup for getting a job, but also surviving the job. I know I answered your question more probably based on a job hunting perspective, but these do help you in the job as well, because in the job, you are dealing with different people, different personalities, different supervisors. You're dealing with cross-functional teams where you want to learn how to be proactive to reach to other teams. Even though that may not be your job requirement, you still should do it because it helps you grow and helps the company grow and so on. Yeah, great question, thank you.
Any other questions?
Thank you, Dr. Tareen. Just one question about the FDA. I mean, you mentioned about how they need to catch up to what you're doing. What do you think, how would you approach that?
How, for them to catch up, right? Yeah.
Yes.
Yeah, good question. So the question is around how do we make sure the FDA catches up as well, because it's not just their responsibility, it is ours as well. So these organizations in the FDA, they consist of scientists like myself who probably came from a biotech background and then chose to work for the government, like the agency itself. And the best thing that I see them do is they come out to conferences. In conferences, you will see a lot of people who work at the FDA who are there learning about the latest science. And I try, and I tell this to my team when you go to conferences, is talk to them, make sure you introduce them so that there's going to be continuing dialogue. So enabling continuing dialogue at a conference setting works really, really well. And then also helping them understand your science work really, really well. And the last part, I believe, is as the agency continues to hire people into their team, they should hire them based on certain foundations, meaning, back to the previous question, are you somebody who is committed to enabling these new therapies to come to patients? How will we find out from an interview whether or not you are that person who is committed, and keeping patient safety, of course, still at heart and in mind? So I think through this three-way approach, hiring the right people with the staff, also allowing the staff to change. One of the reasons I think tech and biotech works really well is because there's a lot of job circulation, and people get fired, people get hired, there's performance reviews, etc. I'm not sure, but I don't anticipate the government agencies to have that strict of an approach. And I think if they incorporate that into their year-end reviews, into their hiring, by making sure they have the right people committed to bringing these therapies to patients and making sure they continue their education, through conferences and keeping open dialogue, I think they will catch up. And as I mentioned in my talk, they've done a lot already to make sure this happens. Yeah, thank you.
I have a question about viruses.
Yes,
So
my favorite topic.
Yeah, it's palpable, your interest. As someone who doesn't know anything about viruses, I didn't know the extent of what they could do. So this was very interesting. If looking toward the future, like if viruses were to thrive in your wildest dreams, you could see viruses doing this. Do you have ideas or hopes for viruses?
Yeah, hopes for viruses. I would love them to survive and thrive and not really bother us too much. And we already, I hope with the examples I gave today, we're already utilizing them for the purposes of generating cures. There is some topics that I get asked a lot is, for example, with global warming and ice melting, people are worried about viruses that we haven't been exposed to, that we might find out of. That is partially true, but let me elaborate on one thing. So ice will have a lot of trapped organisms, single-cell organisms, protists, and so on. And there will be a lot of viruses that infect those organisms because viruses practically infect every organism out there. They don't cross-infect, so a virus that infects a bacteria doesn't infect human cells generally, but every living cell will probably get an infection from a virus at some point. So those viruses won't infect us because they evolved to infect those organisms, but one thing that could happen, and I'm waiting for a news release of this one day, a friend of mine, his name is Neil, actually I'm going to have beers with him tomorrow, I think. He's a really good friend of mine. He worked at the CDC for his postdoc, and he worked on the 1918 Spanish flu. Have you all heard about this? This is this famous, especially during COVID, we heard about it. Oh, there was this flu 100 years ago. And it's called Spanish flu for the wrong reasons. It was actually in Kansas, but it's called Spanish flu because Spain was a neutral country during World War. And they actually announced the infection when other countries didn't as a strategic war reason. So it became known as the Spanish flu, but it started in Kansas. Anyway. So the Kansas virus, all right, that sounds like the China virus that they kept talking, which is right. That's the wrong. So viruses shouldn't be named based on the country because chances are it probably originated somewhere else and so on. Now, what he did is they worked on this virus. Where did they get this virus? Because this virus was 100 years ago. It was from a permafrost sample where in Alaska, I believe, Alaska somewhere, there was a body that was frozen in the permafrost that thawed, and it had died from the Spanish flu. And you can imagine in a cold place, even though that person died and the virus was in them, you can probably isolate, even if not a live virus, you can probably isolate the genetic sequence of the virus and recreate it in the lab. So I love talking to him about this stuff, which is why he's one of my favorite beer buddies. And he tells me examples of how, when they did this work, first of all, imagine being that scientist right next to that body, and then in the biosafety cabinet where you're supposed to work with the right attire, you isolate the lung tissue. And then imagine in those little tiny liquids I keep talking about, you pipette, and then the computer shows these graphs of genetic sequences, the A, G, C, T peaks, which is that flu has RNA, so it would be an RNA sequence. And then imagine from that RNA sequence, you can now resurrect the 1918 Spanish flu. Now, they don't do this in just to their own will. They have to get government approval. It has to go through a very secure chain of approvals and very strict requirements. So they did this, and they studied this in ferrets. Ferrets are these cute little furry creatures, and they are very good virus models because unlike mice or rats, which tend to be inbred, so you buy mice for research and they're inbred, so they don't really represent a wild organism. So ferrets are good for that because there are wild ferrets that are used for research. And in these ferrets, you can do these studies. And same with coronavirus as well for COVID-19. They did studies in ferrets as well because the pathogenesis mimics the human pathogenesis. So they did all these studies. So anyway, of course, I'm not wanting another pandemic to happen, But I would be really curious, when people ask me the permafrost question, I tell them, you don't have to worry about amoebas or bacteria or protist viruses, but if there is a mammal in there that died of some virus, and if you somehow handle that mammal, either as a pathologist, as a scientist, or just as somebody who's hungry, maybe, you want to be maybe careful and let the experts do that. And because, as in the 1918 example, people have used those permafrost samples to isolate very bad infectious samples, yeah. Thank you.
Do you have time for one more question? Is this
Yes,
fine?
yes, I've got time, so please ask away.
So I was just on an aside that I'm an entomologist and a child of the 80s
Very
as well.
cool.
And one of the reasons I became an entomologist was also because I watched scary movies
Yeah.
with large insects, and I thought that was such a cool idea. And so maybe we just need to have film fear festivals and make more
That's
STEM
so
people
cool.
out there. But instead of all this other stuff, we'll just play horror films for students. That would be awesome. I was just going to ask about this sort of T-cell therapy approach. I noticed when I read about it that there's usually a number of diseases or cancers that are candidates for it, that there's a lot of research going on. And so my question is sort of two-part. Number one is, are there certain diseases or cancers that are particularly good candidates and others that for some reason don't work with this therapy, and that's why we don't hear about them? Or is it like a funding thing, right? And it's really about where the money is. And then the second part is about accessibility to gene therapy. Is that something that, because there are trials, et cetera, they're really open up to a diverse array of people who can actually utilize this resource and potential cure? Or is this something that is still really only accessible to people with money and access to these types of therapies?
Yeah, great questions. So we can't cure every cancer today. Blood cancers are a good target for these cell therapies because the cells are given back to the blood and the cancer of problem is already there. Now, in blood cancers, there are subdivisions of cancers from leukemias, lymphomas to myelomas and such. Some of those, even though they're blood cancers, they originate in the marrow, and your chances of curing them with cell therapy change depending on the type of cancer. So often, when you read about this, pay attention to what type of cancer is it approved for. Our drug was approved for diffuse large B-cell lymphomas, DLBCL. There are other leukemias like ALL, acute lymphoblastic leukemia, that is a lot harder to treat. And so for biological and clinical reasons, and the way the field is trying to get around this is it's never just the cell therapy alone. So often our patients are treated with something called preconditioning, where they're given chemotherapy 1 to 2 weeks prior to getting the cells. That preconditioning is there to deplete certain cells in their blood to give this now cell therapy a chance to thrive. So the approval protocols are often in the context of something else and never by itself. So even there, blood cancers, it only works with certain blood cancers, not all, and never by itself, but in context with other treatments. The biggest target now is solid tumors. So solid tumors are the other kinds of tumors, the brains. There's a new company in Seattle called Brainchild that is founded by a good friend of mine who's a professor at Seattle Children's, and they're going after these very difficult to treat pediatric brain tumors. That's a solid cancer. Lung cancers, melanomas, and so on. All these solid tumors have really amazing therapies that are new, such as immunotherapies. There are these antibodies, for example, that are newly approved for some of these solid tumors like melanomas. Patients get them, and that antibody is meant to stop your immune cell and your tumor from touching each other. Why? Because tumors have evolved ways to silence your immune cell So these antibodies are given to block that silencing so that now your immune cells can actually kill the tumor. These are amazing therapies, won Nobel Prizes, and there's also a great documentary about them. I'm forgetting the name, but it's about immunotherapy, and it stars one of the Nobel laureates, Steve. Anyway, I forget his name too, but great documentary. Because it's about the whole story of how his research went from academic research to talking to companies, to convincing companies to, anyway. So solid tumor is the next frontier for cell therapy. Right now, it just doesn't work because if you give the cell therapy by blood, it has to go to the solid tumor. Okay, well, can you give the cell therapy as an injection directly to the site of tumor? Perhaps. But if it's already a localized tumor, you wouldn't need cell therapy. You could treat it with radiation or surgery anyway. So cell therapy is often used when the disease has spread. So how do you make sure cell therapy, it has to touch, because it relies on contact. Those receptors we add rely on contact and proximity for killing. So that's why solid tumor is one of the next frontiers. And companies, including one in Seattle as well, many in Seattle, of course, not just one, are working really hard to figure that out. So that's the answer to the first question. The second is accessibility. This is a really interesting topic because in my previous company, Juno Therapeutics, where we got the FDA approval drug, they hired somebody and they shipped them, I think they hired them from Washington, D.C. They hired them to be the policy influencer at D.C., to talk to insurance companies to convince them that they should cover this therapy, to talk to lawmakers and policymakers to convince them that these therapies are really life-changing and everybody should have access to them. Now, you're correct. They come with a price tag. You often read the news of gene therapies that are record-breaking price costs at $1 million, $2 million, $3 million. These CAR T-cell therapies that I mentioned I think are about $500,000, if I recall. And there is insurance coverage, et cetera, but you're right. Why are they so expensive? I think there are several reasons. Number one, it takes years of research to get this done. My team alone, we're about 12 people at Sana. Our yearly expenditure was like 12 million, and that doesn't include salaries. So we were spending, and I know this number because every year I have to do a budget calculation and such. We are spending $2 million on disposables, on reagents, and we're just a team of 10 to 12 people, not including salaries. And then our company, Sana Biotechnology, was about 500 at its peak. So you do the math, and then you have a range of, so it takes a chunk of time. And the main reason, time and money, the main reason though they're expensive, I think, is academic research always happens with tax money. Breakthroughs happen in academic research almost all the time. Breakthroughs don't really happen in companies. What companies do is they take that academic research, make it, they industrify it, make it accessible to the masses based on regulations. For example, we had to build a GMP plant in Bothell for millions and millions of dollars because that's what the regulations require us. You can't just make this in any lab. And getting into people. You have to test it through very rigorous quality testing and so on and so forth. And that's a good thing. And only then will patients have access to this. So for that biotech life cycle to begin, you need investments. Investments come from VC firms. If you look at all these Seattle companies, you'll see VC firms like Arch Ventures who put in, one website I read every morning with my coffee is fiercebiotech.com. It's accessible to everyone. Fiercebiotech.com is a great website to see, oh, so-and-so company raised 50 million round A for their new therapy, this, that. Sana Biotechnology, we raised 800 million, for example. So, and guess what? We ran out. So we didn't run out completely, but there were massive layoffs. 2023 was a big year for layoffs in biotech. My team and we got laid off as well in October. So I'm talking to you jobless right now, but hopefully the next thing is going to happen soon. So the reason we were laid off is the R&D was done. Research and development our job was done, because now it's all in the clinical trial. They have to save the remaining money to run the clinical trial, which is very expensive. A clinical trial alone costs about 50 million to have 10 patients in a, so I think it's for these, I honestly don't know, and I would love to talk to economists to understand this better. I honestly don't know how we can break this cycle, but I'll give you an example. When I talked to, so I was in Turkey over holiday break, and I was talking to universities about, they don't have any CAR T-cell therapies approved in that part of the world yet. I was talking to them about how do we bring these therapies here, and I was trying to do the financial calculation, and I think you still fall into the same trap of, okay, it's probably going to be cheaper over there, but I still need to build the space. It probably is going to be cheaper over there, but I need to hire the people. It's probably cheaper there, but I need to get all the machine and equipment. So you still end up needing VC money. Maybe it's for a lower cost, but it's still needed. But I do believe that the costs, they have gone down in a lot of therapeutic areas where people are figuring out how to make these accessible. One example is, we want to give this therapy now directly to the patient as a virus, not in the lab. So that's going to cut all the costs in the manufacturing of that 2 to 4 week manufacturing cycle and so on. So long answer to the two excellent questions. By the way, quality assurance and quality control is a big part of our life. I don't do that work, but I have amazing colleagues who do this. There's a fascinating book called The Cutter Incident. The Cutter Incident with a C, C-U-T-T-E-R. Cutter Incident is about one of the polio vaccines, and there was a lab called Cutter Labs, and they released polio vaccines where they hadn't inadequately inactivated the virus. So a bunch of kids back in the day got polio and got paralysis from this vaccine. So of course, it was a game changer in how QA, quality assurance, and QC, quality control, should be done in the healthcare setting. And there are fascinating stories like this of how the industry just has gotten better and better and better, and will continue to get better over the years with regulations and with these controls in place. But all these also now cost a lot of money. All these tests have to be run and so on for the right reason. So Cutter Incident is a great, fascinating book about a really successful story of the Marchadimes and the polio vaccines, et cetera, but also this Cutter Lab incident just reminds us how important quality control is for this. Yeah. Thank you, great question.
Okay, we have one last question, and this is for the students who are here. I appreciate that students came. So, Semih, what advice would you give our students who are interested in being researchers, scientific researchers?
Yeah, thank you. I think we already concluded that watching horror films is a good thing. Right, from the insects and the viruses, and joke aside, I do think imagination does help. Because I grew up in Turkey, I came from a very strict education system, and STEM education is really strong in Turkey as well. So Microsoft, for example, hires a lot of people from Turkey, from India, and so on for that very reason, because we tend to freak out about math at much younger years than I noticed education here does. And I don't think that's a good thing either. So it is very important for students to not lose their creative side, because that creativity comes into play. When you're creative, it doesn't have to be music or movies, no, none of that. It can be any type of creativity, but it allows you to think outside the box. It allows you to articulate your point to your colleagues around you in a way that if you are able to influence, then maybe you could convince them to try something out. And trying something out is a big deal in biotechnology. If people didn't have the courage to try things out, to have buy-in to try things out, and to accomplish all these things, I think you need creativity. But back to the teacher's wonderful question earlier around, you need to learn that skill of articulating your knowledge and articulating what your plans are and why, to get buy-in. The earliest you do this, the better. Because right now, this is how I learned this. I learned this after many, many, many years in the job. And it would have been best if I knew a little bit more about it going into the job earlier. It didn't hinder my development, I don't think, but I do think that advice would be great. Yeah.
Thank you very much.
You're
So,
very welcome.
thank you, Semih. Thank you, all of you, for being here.
Thank you. I really appreciate your time and the great questions. Thank you. And the blue screen, I hope, kind of gave me a nice little silhouette in the back. Yeah? All right. Thanks a lot. Take care.
Thank you. We hope to see you at our next event, which we're trying to schedule for March. So thank you all. Thank you so much.
You're very welcome. I hope that was helpful.